This is the last lecture. I'll post it on YouTube. It's Evidence for Evolution. As you know, there's within certain segments of society, there are people who call evolution only a theory. So we're just going to cover some of the lines of evidence that suggest that populations evolve and life is billions of years old and that sort of stuff. There was a lecture that I posted earlier, I'll post it again, on Evidence for Evolution by a guy named Jerry Coyne, who is at the University of Chicago. And it's about just under an hour. You might want to watch that as well. We'll be covering some of the same topics. Okay, so some related concepts. A fact. Fact is a known truth. Of course, in science, there are facts, but because the evidence is so strong. But remember, everything in science is provisional, even though we've reduced the uncertainty to almost zero on certain things. A law is a general statement about how natural phenomena behave. A hypothesis, of course, is a testable proposition or explanation. It has to be falsifiable. And a theory is usually a larger thing than a hypothesis. And it's got more evidence, and it's been tested over and over again and has never been disproved. Versus a theory, in the lay sense, of course, that, it's only a theory, which is mere speculation. So, again, I've already marked this up, but I'll make it yellow this time. Terminology. So we often come up with questions about, we observe something and we wonder why we see what we see or why we detect what we detect. And so usually you ask a question. What do you do when you want to understand something about the natural world? And as you know, it's been hypothesized that the moon is made of green cheese. But I'm going to change that, and it's going to be yellow now. There's a lot of yellow cheeses out there, so I don't think we can have too much problem with that. Well, first of all, hypothesis is a tentative proposed explanation or answer that describes some process to your question. In other words, it's a proposed explanation. What do you think might be going on? And hypotheses are a dime a dozen. They do have to be testable, though. In other words, you have to be able to disprove them. And we really don't go for proof in science at all. I know it seems like we do, but we actually try to disprove things. And the more you try to disprove, let's say, a proposition or a hypothesis, and you can't, the more evidence, especially if the testing is more and more rigorous, the more likely it is that it is representative of some real thing about the universe that we live in. But it's still only a conjecture, and it's what might be going on. Your hypothesis, it could be an informed guess based on current states of knowledge, okay? But you could even propose something that's just completely crazy, as long as it's testable, in other words, as long as you can disprove it, it's a valid hypothesis. So, if you remember, initially in class, when we talked about hypotheses, I dropped my keys on the floor and I suggested that gravity is because there's men or people in the center of the earth that have metal heads, and that these metallic headed people create gravity, even though gravity is, of course, a different force in the universe aside from electromagnetism. So there's 4 different forces. You don't have to know this, but electromagnetism is one, gravity is another. And of course, it was easy to refute because I dropped it. And then one of the predictions, and this is where things change, a hypothesis makes predictions, and those are the things you actually test. And so, if in fact gravity was like electromagnetism, it should only work on metallic objects. And those are the things that should fall to the ground. Well, my keys certainly fell to the ground, but none of us are floating off into space. And so, obviously, gravity also affects us, and we're not metallic objects. And so, the prediction, in terms of the metal-headed individuals who create gravity, has been refuted. Okay, we didn't even really have to do an experiment. We just had to make an observation that completely discombobulated that argument. Okay, so we have this, we'll go back to green. And, okay. Okay, so hypothesis or prediction. Well, a hypothesis, remember, is an explanation. And it makes certain predictions that are falsifiable. Testable, disprovable, which means falsifiable. So you can disprove it. We don't actually ever prove anything. We just test the predictions, sometimes with more and more rigor, to the point that if we cannot disprove it, then that's provisionally accepted as truth or a representative of reality. Okay? Because there's always the possibility that new information and new testing will come about that will disprove that hypothesis. But some hypotheses and some theories have such strong evidence that the uncertainty around whether they're true or not is so small that it's almost like there's a fact. And we treat a lot of those things like that. So, and furthermore, we actually collect data to falsify that hypothesis, in other words, refute it, instead of looking to support, because we want to avoid something called confirmation bias. In other words, we want to avoid fooling ourselves into thinking we know what's going on, okay? Because if you come up with a great idea, it's really nice to find evidence that your great idea is, in fact, true. But there's other things that go with that. Sometimes, you think, oh, I'm really smart. I've come up with a great idea. And if I can show that it's true, then I am really smart. But you don't want to do that as a scientist. You actually want to find out if your explanation represents objective reality. Okay. So, and a lot of times instead of one hypothesis, sometimes we make a number of hypotheses, competing hypotheses, for why we see certain things. And they should make divergent predictions so that we can actually separate out which one has the most support and which ones we can cut out at the knees right out of hand. And so if you find a hypothesis that's not supported, in other words, you've refuted it, then we've refuted it and it's not correct. Of course, if you test it in a way that you didn't really test it, then you might not have. So you have to be very careful in making sure that you actually tested what you say you're testing in terms of the explanation. Okay, experiment or observation. If you remember, there's a difference between experiment, where you have one variable, let's say in a lab, and held everything else the same. You have replicates, let's say you have a drug that you're testing versus a placebo. You have each group has 100 people randomly assigned to one of the two groups. You give the drug. They have, let's say they have, it's a new antibiotic or something. You infect them with a bacteria. Well, you can't do that with people, but let's say it's animals. And then you see if the antibiotic works and if there are any bad side effects, right? Okay, so, but we can't always do that. Sometimes we can certainly make hypotheses, explanations, in other words, that have testable predictions, but instead of setting up an experiment, we have to rely on observation. So for historical sciences, whether it's cosmology, astronomy, evolutionary biology in a lot of cases, especially with the fossil record, geology, we can't actually run experiments. We can run let's say mathematical models. But if we had control of time, we could go back in time. Let's say we want to know if an asteroid killed the dinosaurs. Well, it'd be nice to go back in time and see if an asteroid actually did it. Or we could, let's say, replicate the Earth 100 times and then hit it with asteroids versus, in other cases, let's say, causing damage because of volcanism or something like that. Let's say different hypotheses, and see if we would end up the same sort of extinction rate and adaptive radiations that followed. But we can't actually do that, so we're stuck with making hypotheses. Those hypotheses make predictions, and then we actually have to test them against what we actually observe instead of controlling the variables. But that's just the way it is. It doesn't mean the inferences are, let's say, well, they're a bit weaker, but that's all we can really do. And with enough independent lines of evidence, for example, using, let's say, geological layers that have been dated with various methods, finding certain fossils in some layers where they belong, let's say. In other words, you wouldn't expect to find humans and dinosaurs in the same layer, right? Just modern humans didn't really arrive on the scene anatomically, modern humans, until about 300,000 years ago, and of course dinosaurs were wiped out except for the birds at 65 million. Now, of course, we could go, in this case, this is a stupid thing. I'll bring a piece of green cheese and we'll examine it, whether it is like green cheese. And of course, we found out that there was no green cheese, so it was all rocks. So, in other words, you can actually measure things. We didn't find any example of green cheese, but we found different kinds of rocks, and there's some other things we could certainly find out. Are the rocks on the moon? One of the hypotheses is that the moon was once part of a larger Earth, and that it was struck by some other large object in the early formation of the universe and knocked a big chunk off, and that became the moon. Now, if that's the case, we can make a hypothesis. That's the hypothesis. Versus, the moon was another large object that was created independently of the Earth, and perhaps the rocks on the moon would not match the rocks on Earth, versus the hypothesis that the moon was actually once part of the Earth, and then the rocks, the kinds of rocks and the chemical nature of them would be similar. As you know, data or observations often suggest new questions and hypotheses, or experiments where you reach a conclusion. Let's say the evidence supports this versus that hypothesis. That will often generate new questions and new hypotheses, so that usually the first bit is just the beginning of exploring mechanisms for why we see things the way. So a theory is a little different. First of all, to become a theory, it usually has to provide a bigger explanation for some broader phenomenon than just an individual hypothesis. And theories actually often have different hypotheses that make them up. But hypotheses, in a sense, are like a dime a dozen. You can come up with any of them as long as you can falsify them. They're valid hypotheses, and then you can either blow them up or they might have some merit. But in theories, scientific theories are a different animal altogether. Not only are they made up of testable hypotheses, but those hypotheses have not been falsified, and the theory itself, after rigorous testing, has never been falsified. If it has been falsified at some future date, then it's either tossed out and replaced or modified. Okay, so a good example is the theory of gravity. The first theory of gravity was by Newton in the 1660s, and he proposed a mathematical model that accounted for gravity. And then subsequently, with the optics at that time and later optics that were better, they made measurements to see if, in terms of planetary motion, whether that coincided with the predictions of his mathematical model of the solar system. And his model worked very, very well, but there were some discrepancies. There was something a little wrong with Mercury. There's some things that just don't quite add up, but it's a very good predictive model. In fact, it's still used in terms of space travel and whatnot. But in 1905, actually, in 1915 or around there, Einstein came up with his own theory of gravity that was completely novel. It was based on relativity, general relativity, in terms of how to explain what gravity is, and it made some very, very strange predictions that are all being borne out. And in fact, really, Newtonian physics is really not compatible with Einsteinian general relativity. But we still use it for practical purposes, but our real understanding of the universe in terms of the theory of gravity or general relativity has actually replaced it. But that's how science works. You keep testing things, and then eventually new knowledge comes along that raises questions about what your model or hypothesis or theory predicted, and there might be some discrepancies, and you find out someone comes along and thinks about a new way of looking at it. So scientific ideas really don't attain the status of theory, and it's not like, oh, it's only a theory. No, a real scientific theory are well supported by multiple lines of evidence and many, many, many independent experiments and observations. That doesn't mean that it cannot be overturned. So after an idea, let's say for some general phenomena, then there's usually ongoing research to focus on extensions and implications and whatnot. Whether anything that any let's say theory that humans come up with is actually a perfect description of reality remains to be seen, but this is what we have. So theories are pretty rigorous animals that requires really, really rigorous requirement, has really, really rigorous requirements. And even then, they can be overturned. Okay? But usually you don't overturn a theory by one errant observation. You go back and, for example, a couple of years ago, some physicists claimed that they measured something that, I think it was electrons or something that went faster than the speed of light. In other words, they sent a signal from one place in Europe to another place a couple of hundred miles away, and supposedly the photons, I think it was light cables, supposedly it got to the location quicker than light is supposed to, which is constant. It's a constant. It only goes so fast, about 300,000 kilometers a second. Okay, but they of course said, well, it probably is a problem, and they went back and of course they found out that it was their equipment, and if they had actually measured something that went faster than the speed of light, it would have overturned special relativity and general relativity and our whole view of the universe. So Sherlock, he here came up with Colonel Mustard hit him with a lead pipe, right? Well, that's actually a hypothesis. It's too bad that the criminal justice system actually doesn't often use the scientific method in their work, and that's why a lot of people get locked up who turn out later to be innocent. Okay. theory of heliocentrism. In other words, um, previous to Copernicus and Kepler, it was thought that that we have a geocentric universe. In other words, the Earth is at the center of the universe and everything revolves around it. And it's not hard to understand why people would believe that. And it wasn't until the theory of heliocentrism was proposed. But then, since then, of course, that's just the solar system, and the universe is now, we understand, is much, much bigger and older than that simple theory of heliocentrism. So, theory of relativity, relationship between mass and energy with implications for space-time. E equals mc squared. E is the energy. Mass is some measurement of mass. And then the speed of light squared. So, finally, we come to the theory of evolution, which has been something of a contentious endeavor. Now, of course, in addition to thinking previous to the 15th century that the Earth was the center of the universe, it was thought that nothing changed, things were immutable. In other words, the Earth was actually only about 6,000 years old. Geological processes is all recent stuff. When you find seafloor uplifted onto some other place, it's an example of the flood, that kind of stuff. And then there were other parts to that. And finally, it was the idea that it was at least thought that God created species and all living life specifically, and that's why we see these adaptations that show the benevolence of God in terms of the Judeo-Christian Muslim idea of the cosmos. But evidence started showing up, like fossils and things like that, and evidence, it seemed like, that there were related species that seemed to be descended from others, and so eventually ideas about the fact that organisms change, lineages and populations change through time came to the fore. And then it wasn't really until Darwin that a mechanism was actually proposed, and namely evolution by natural selection, along with other mechanisms that were discovered like drift and things like that. So there's a lot of evidence that turns out for this theory now that we have of evolution. So modern evolutionary theory isn't just what Darwin proposed, but it's been built on since then. And it certainly explains a lot of what we see in the natural world. So, what we're going to do now is explore a little bit of some of the evidence that supports that view, and then perhaps contrast that with a view that, that, let's say, a creator or intelligent being created everything, including man in God's image or whatever. Okay, so we've got this tree of life, diversification through time, and some key pieces of evidence include homology. If you remember what homology is, it is traits that are inherited from a common ancestor. So tetrapods, including ourselves with 4 limbs, we're vertebrates. We're a class of vertebrates that have 4 limbs. The bones in those limbs, even though the limbs may look radically different, let's say between a bird and a walrus and an elephant, the same bones are used, and it's just that evolution by natural selection has shaped that morphological diversification. And it's not just anatomy, it's also physiology and physiological processes. It's also genetic homology. In other words, you see the same sequences over and over and over again when you sequence organisms. So we have many of the same genes operating in us as bacteria do. There might be some variants, but in general, those are the same sequences. There's also other bits of evidence, vestigial traits. If you remember, vestigial means traits that in an ancestor had a function and have this since then lost that function. And things like the vestigial traits where they had something had a certain function in an ancestor and then might have maintained that function but also developed new functions as well. And then the idea of transitional forms, namely like the missing link. People for a long time were looking for the missing link between chimpanzees and humans, right? Well, we've since found the fossils of many ancient hominins, some very old, like almost 5 million years old, that might possibly be on the human lineage. Others more recent, like Australopithecus, and then others even more recent, like Neanderthals. We see this again and again in the fossil record. But you have to understand something. The fossil record, first of all, is very incomplete. And you might say, well, why? And we'll get to that in a little bit. So, question for today, what is evolution? What's the evidence for evolution? And how did Darwin come up with the idea of not just natural selection, but evolution by natural selection, which is the only mechanism that results in adaptation to the current environment. There was a theoretical population biologist, evolutionary theorist named Theodos Dobzhansky, and he said nothing in biology makes sense except in the light of evolution. Well, first of all, before there was evolution, or you can still do medicine without evolution, in terms of mechanistic, ways to cure certain diseases. But there are other features, of course, let's say in medicine that only with evolution will you actually understand why they're there, like the antibiotic resistance, bacterial antibiotic resistance, or legacy traits that actually cause disease, genetic diseases that are the result of various mechanisms that lead to a less than optimal design, things like that. So, in terms of what is evolution? Well, we know, first of all, that a change in the frequency of alleles in a population over time. And that's caused, of course, by the four forces of evolution. And then genotype frequencies, if you add that, then it's also non-random mating and sexual selection. Okay, so here we have frequencies of alleles in a population. We have time 1 and time 2. As you can see, we've got a lot of reds, and we also have a lot of greens. Okay, it might be the same, let's say 50%, 50%. And then if we look at this population in terms of these two alleles, red and green, red squares, green triangles, we see in times 2 things have changed. There are now greater number of red alleles in the population than compared to the green triangles. This could be the result of a number of different things. It could have been drift, but probably not. It looks more like kind of selection against green triangles in the environment that this organism is found in. Okay, so basically what happened was a bunch of these green triangles got wiped out, and a bunch of these red squares had babies and multiplied. And so now you have not only fewer green triangles, but greater many more red squares than were before. We know what an allele is by now. An allele is a variant of a gene. We've been mainly modeling it as, let's say, 2 alleles for each gene. And of course, since we're diploid, we do have 2 alleles for each gene. Sometimes it's the same allele, mother and father. Sometimes they're different alleles. But we're limited to those 2, even if there are many alleles for that same gene in the population, which is often what happens. So in terms of the whole population, you might have, in some cases, 1,000 alleles for one locus, but each individual diploid organism is only going to have 2 of those. They might be the same 2 or different 2. So cystic fibrosis is a genetic disease. It's caused by a deletion of CTT, in other words, cytosine, thymine. And that deletion that codes for that protein, you end up with a non-functional protein that causes problems, namely mucus in individuals with cystic fibrosis tend to be very thick, and in the lungs difficult to clear, and elsewhere, like in the gut, causes problems with digestion. It's actually a recessive allele. The worst effects are 2 copies of that allele, that mutation, deleterious mutation allele. And that's only likely to happen in, let's say, some inbred populations. So it's mainly found in Northern Europeans and some other groups at higher frequency than other populations. So remember, alleles or genes don't code for traits, they code for proteins that then make those traits. And it can be all kinds of different traits. It can be anatomical, developmental, it can be behavioral, physiological, all that kind of stuff. Okay? And it might be that certain individuals who, let's say, with a cystic fibrosis allele, selection, because of the changes it causes in the protein, selection acts against that protein and therefore that allele in the population and removes it by purifying selection. So in other words, selection is acting on the individual with that unfortunate mutation. And then, of course, and what evolves actually is the frequency of that mutation or that allele in the population over time. And so with cystic fibrosis, we would expect, of course, that selection would remove individuals with 2 cystic fibrosis recessive alleles. It lowers their fitness, and they tend, if they do survive a little older, into their 30s, let's say, they probably will not have too many offspring. And so there's pretty strong selection against that. Unfortunately, there might be a situation that's similar to sickle cell, where cystic fibrosis allele, a single, might protect against typhoid fever, but that's still up in the air. So anyway, here we have populations, anywhere from bacteria, to bees, to penguins, to these damselfish, I think, these pond turtles, and these gazelles. So we know that populations evolve, individuals don't. Their cells might change, like in cancer, and that's actually populations of cells evolving. So you have mutations that allow some cells to reproduce more often than others, and they quickly overwhelm the system. Even if it eventually kills the individual that cell population is in, those cells proliferate. Okay, so in the same way, populations evolve, namely, alleles that, in terms of natural selection, are favored. That means confer some trait that has advantageous and beneficial properties. You would expect that allele to increase in the population generally in terms of evolution by natural selection. Okay? Now, remember, you can also have changes in allele frequencies just by random events, like mutation is random. Some alleles might be beneficial. Most will probably either have no effect or be deleterious or bad, and those will be removed quickly from the population. Okay? So, and those alleles code for different proteins and therefore different traits. So, genetic drift is another example. Migration can either be good or bad, depending. But none of those three forces actually consistently generate beneficial or adaptive evolution. Only evolution by natural selection does. So, we look at evidence for evolutionary change. And we're going to go through the fossil record, comparative anatomy, comparative embryology, in other words, development, molecular data of living organisms and a few organisms that have gone extinct, but we've recovered their DNA, and then evidence for evolution in action, like antibiotic resistance and things like that, and actually more than that. Okay, so fossils are the remains of plants, animals, and fungi. Any living thing has been fossilized, including viruses, bacteria, things like that. Okay, and they're often found in mineralized form in rocks, sometimes in amber, which are waxy stuff from plants that entomb, let's say, insects. Some of them are impressions like dinosaur feet, that sort of thing, footprints, things like that. There's all different kinds of fossilized remains. You usually find them in sedimentary rock. That means deposition of things on top of each other. So let's say you have a seafloor, animals, let's say fish or whatever, die, they float to the bottom and they get covered up. On the right are trilobites, one of the longest organisms with a very, very long tenure on the earth, but they're all now extinct. So extinction is a big deal. So layers of rock contain fossils, new layers cover older ones, and it creates a record over time, right? So the deeper you go, the older the rock. But you have to remember there's also things that are going on in geology. There's uplifts. So you can occasionally, like if you go in the Andes, you can actually find whales up at 19,000 feet. that area was once sea floor that has now been uplifted into a mountain range. Now, we can also date fossils. So remember, there's strata. Okay, so there's radioisotopes like uranium, 235 and 237 and 238. Uranium decays into lead and, well, first it decays, I think, into 235, or I forget how it goes. But there's different kinds of radioactive isotopes that decay at different rates. So like carbon-14 is a radioactive isotope of carbon, and it has a half-life of about, I think it's something like maybe 20,000 years. In other words, or less, probably 8,000. So it's only good until about 60,000 or 70,000 years old, and then there's too little left to actually date it. There's other isotopes like uranium, and there's two different things that it decays into. One's a couple of million years old for a half-life, and the other is billions, and so it's often used to date very old things. You probably wouldn't want to use that for something that's only like 8,000 years old. There's been too little decay, but so you'd use something like carbon-14 instead. And so there's these different isotopes you can use, and it's called radiometric dating. Okay, and it's relative dating in terms of position of rock layer with respect to a known layer of rock and how old it is. And the amount of parent isotope declines with time at a predictable rate, and we usually see it half-lives. So let's say at time 1, there's now half as much of that radioisotope that was there. At time 2, there's now a quarter. At times 3, there's now an eighth, a 16th, and so on, until there's so little left that you really have nothing to actually date beyond that. So we'll say here's time, and up at the top you might have a strata at the very top, and then that you might have another strata, and another strata, and another strata, and then a large discontinuity. We'll make it pink. There was strata here, but it's all gone. And then underneath that, you might have some more strata examples. Okay, so this is time, obviously down here, older, younger. So if you have a reference rock layer that you know exactly how old it is, then you can use relative dating in terms of other rock layers to it. And this decay happens at a predictable rate depending on the radioisotope. So in other words, radiocarbon dating, like I said, is only good until about 60,000 years. I'm not sure what the half-life is. I think it's about 7,000 or 8,000. Okay, so here's kind of an evolutionary timeline. We have something right down here is the oldest, 4.5 billion years. There is very little remaining from that time period in terms of rocks. Liquid water didn't really show up on Earth until okay, so formation of the solar system, a little over 4.5 billion years. The moon forms, very violent early in the solar system. Lots of things crashing into each other. A disk is forming out of primordial hydrogen gas, along with the sun, of course. Things cool down. So about 4 billion years ago, there's now liquid water on Earth. Before that, it's really too hot. 3.8 billion years ago, first oceans. There's still heavy bombardment, collisions with large asteroids and other bodies from space. It finally starts to end around 3.8 billion years. So that's the end of the Hadean eon. Then the Archean eon goes from 3.8 to 2.5 billion years. During the Archean eon, we see the origin of life at some point. We don't know exactly when it happened. It might have actually happened all the way up here or earlier. First evidence of photosynthetic cells. The thing is, there were probably chemosynthetic or cells that instead of using light, they used elements like sulfur to generate carbohydrates and things like what plants do, but it's a chemosynthetic thing. There's still organisms that do that. If you go to these vents deep in the ocean, there's no sunlight, but there's all these chemicals coming out, and it provides the basis for food chains starting with bacteria that feed on this stuff, and then other things that feed on them. First evidence of oxygenic photosynthesis, in other words, photosynthesis that produces oxygen. The first cyanobacteria are about 2.5 billion years old that we know of. First rocks containing oxygen, in other words, there was finally enough oxygen that was produced by photosynthetic organisms that it was free oxygen in the atmosphere and the water. Oxygen is highly reactive, so it kind of turns into chemical compounds like iron oxide and things like that very rapidly, gets locked up in rocks. And it's only when there's so much oxygen that being produced by these photosynthetic organisms that you finally have oxygen in terms of free oxygen. And then finally, you have the Proterozoic eon from 2.5 billion years. And you have first eukaryotic fossils. That means with single-cell organisms with a nucleus that you see in the fossil records. The first algae and evidence of sexual structures. Let's see. First lichen-like organisms. And the first animals were all sponges, and up to about almost 800 million years ago. So a long time ago. Ocean completely oxygenated. First bilaterally symmetric animals. First bilaterally, first sponges. Actually, the sponges, it shows here, they're actually older. First sponges. $800 million years ago. Okay. And then finally we have the Cambrian, and the Cambrian was now there were also plants, or the beginnings of plants, and algae and all that kind of stuff. But the Cambrian, there was an explosion of Metazoan animals, sponges, arthropods, all these kinds of different things suddenly showed up in the fossil record. This is obviously over 50 million or 100 million years, something like that. But it seems like it's coincided with saturation of loss of oxygen that allowed multicellular life to originate and then evolve and speciate. There were of course multicellular algae and perhaps aquatic plants in freshwater environments, but they didn't really show up in terms of on land until the Ordovician. First bryozoans, and let's take a look. First fungi, mycorrhizal fungi, Silurian, first bony fish, Devonian, a bunch of arthropod radiation, first insects, also first fishes with jaws, first ferns and vascular plants, first tree-sized plants, winged insects, tetrapods, first seed plants, Carboniferous, first reptiles, the Permian, it was Carnivorous, was Mississippian and Pennsylvanian. So if you go east of the Rockies, the oldest rocks you're going to find, or the youngest rocks, are going to be Mississippian rocks, 330 million years old. We have the first mammal-like reptiles, or first reptiles, first mammal-like reptiles. The first badiomycete fungi, right? First plants with vessel elements. First okay, so right here was, by the way, was the biggest mass extinction ever. Oh, about 95% of all multicellular life went extinct. And then we enter the Mesozoic. So this is all Paleozoic. And this is Mesozoic. Until from here to the Triassic. Okay? And first mammals in the Jurassic, the Triassic. First dinosaurs in the Triassic, first Tyrannosaurid dinosaurs in the Jurassic, the first birds or bird-like reptile dinosaurs, late Jurassic, early Cretaceous, first angiosperms, remember those plants that produce flowers, first centric diatoms, water lilies, first magnolias in the family of plants, first bees and ants, late Cretaceous. Let me see something. First placental mammals at the end of the Cretaceous. And then finally we get into the Holocene, which goes from right here all the way up to the present. And it's a Paleocene, including the Paleocene, Eocene, Oligocene, then the Neogene, so oldest pollen from daisy family plants, first apes in the Oligocene, almost 33 or something million years, 23, 30 million years ago. First orchids, we have the Miocene, Pliocene, Pleistocene, first earliest hominins, in other words, from about 6 million years ago. The Holocene is the most recent, and here we are. You can also see where there's been 5 major extinctions. 2, 3, 4, 5. And then there's also one that looks like earlier in the Cambrian or older. But these are during the modern age, from the Paleozoic through the Mesozoic um, to common times. And there was an Ordovician extinction, mass extinction, Devonian. The biggest one was a Permian mass extinction, a Triassic mass extinction between the Triassic and Jurassic, and then the Cretaceous extinction that wiped out the dinosaurs. Okay, so why are traits and taxa nested in a hierarchical pattern? And remember, so of course, this is all time. Again, time. Modern forms suggest order of evolution. So by hierarchy, we mean younger down to older, and younger already nested inside older. So, okay. And you can show it any which way. Here's, of course, we're most familiar with, is this phylogeny. We have fish. These are all vertebrates. So we have backbones is the oldest trait. From here on, we have 4 limbs, right? That's nested. So all these individuals on also have backbones. Older ones don't have 4 limbs. Then we have amniotic eggs, right? And they're nested. So everything from here on out is all amniotes, including mammals, birds, and reptiles, and dinosaurs, all that kind of stuff. And then further along, like in birds, we've got other traits that show up. We'll say feathers. Mammals, we have fur and mammary glands. Each of these are nested within. And so if we look at this fossil record, we can see in terms of vertebrates, there first were certain kinds of fish, jawless fish, then placoderms, which are now extinct, cartilaginous fishes like chondrichthyes, sharks and rays. There's also bony fishes that showed up. And then finally the first tetrapods right here. Finally, from here on, we've got amniotes, including reptiles there, mammals around here, birds, dinosaurs around there, and you can see how it all works out. So we have these ancestor-descendant relationships through time. More primitive traits are found earlier. Organisms that are younger often have those primitive traits plus new traits that are derived. Here we have time. We have young layers, slightly older layers, all the way down, right? Until we have the oldest layers. There are some layers that are not there anymore, whatever. So you would expect organisms with more primitive traits would be older, found in older strata. Their descendants might still have those traits, but more newly derived traits like synapomorphies would be found in younger strata, differentiating between one group of animals and then a new group that's descended from them. Show that again. So let's say we have animals, basal animals right here, that have certain traits associated with animals. Okay? 3 layers of tissue, or 2 layers of tissue right down here. A little bit older or younger are 3 layers of tissue. All animals from here on will all have 3 layers of tissue. But then here, there will also be a coelom nested within inside of it, right? So first 3 layers of tissue, then a coelom, and then finally we'll say bilateral symmetry, right, in animals. And then all these other things that showed up. Let's say protostome development. Oops. Or some development found in certain protostomes and not another. And then deuterostome development, right? Those are both came out about the same time, I think. And then in deuterostomes, the first chordates, or echinoderms rather, and then chordates. Over here, we have all these different lineages. Let's say an exoskeleton in insects, mollusks that have certain things. So everything is nested within larger, older taxa. So here's the Cambrian explosion. First sponges appear about 785 million years, maybe older, coincided with the oxygenation of the oceans. Little diversity until the Cambrian, and then suddenly we have all these different groups besides sponges, including diploblast organisms like jellyfish and stuff like that. We also have triploblasts. We have a series of a bunch of worms. We have hard-bodied forms. We have soft-bodied forms. We have the first arthropods. Even the first vertebrates show up in the Cambrian. There are no jawed fish yet, but there are vertebrates. But they're really primitive vertebrates at that point. This all was, you can actually find these fossils in certain rock forms. A lot of them were soft-bodied, but they managed to fossilize anyway, like the Burgess Shale in Canada, British Columbia. Okay. Now, one thing that you would expect is to find transitional organisms between one group and another. So, many years ago, people were looking for the missing link or the missing ape. In other words, a transitional species between apes, like chimpanzees and gorillas, and humans. Now, as you know, we've actually found many, many, many different species of hominins that split off from the chimpanzee lineage. And there's some dispute over whether certain of these taxa are hominins or some different line. But from about 5 million years ago, the bipedalism might start to appear, at least a little bit. 4 million years ago, by 3.6 million years ago, there's a lot of bipedal hominins that are walking around like Lucy. The genus Homo finally shows up about almost 2 million years ago. And then subsequently there were other groups like Neanderthals and Denisovans and Ergaster, a bunch of other Homos species. And they co-occurred, some of them occurred at the same time, like the Denisovans, modern humans, and Neanderthals, and maybe some other ones were living in Africa. All these different species were closely related to each other, but separate species. For some reason now, the only ones left over are modern Homo sapiens. Not sure exactly why. There was some interbreeding, though, between them all. Okay, so we also see this evolutionary transitions in the fossil record. So we have here, Archaeopteryx, considered the first bird, okay? But it has some primitive and some derived traits that you associate with birds, okay? Primitive traits. It actually has a tail, a vertebral tail, okay? It also has teeth in its jaw, okay? It doesn't quite have a furcula, so it was able to fly, but it certainly had flight feathers, which were a derived trait, and it had it was bipedal, of course, like all theropod dinosaurs, and it had feet like some dinosaurs too. I don't think it had hollow bones, but it was probably able to fly. And then from then on, things started to change. Eventually, a tail, selection against a tail because of extra weight. Instead of teeth, it was finally, you ended up with the evolution of a beak. There were some bone changes, including hollow bones, so that flight would be less. All of these individuals laid eggs. Actually, carrying around live-bearing young is expensive, or live young, and so it's better to drop an egg, and that way, when they hatch, they can provision the offspring. Okay. Of course, bats didn't do that. Mammals, it turns out, bats are placental mammals, and they actually continue to bear live young. Obviously, by that time there were placentas, and the evolution of, let's say, losing placenta and going back to eggs was not possible. Same thing we can find in the fossil record. Modern whales and dolphins arose from land-dwelling animals. Seen right here. Then some animals like these that were living in kind of estuarian systems finally started, they still had front limbs, they still walked on all fours, but they spent a lot of time in water. Eventually there was limb reduction, and finally loss of limbs, but still vestigial traits. So a remnant pelvis in modern cetaceans. The same thing happened with lizards and snakes. There were also reptiles that went back to the sea. In other words, land reptiles that lost their limbs as they re-entered a watery environment. So we have all these kinds of things. There's also been transition fossils from the Devonian between the first tetrapods. So you have fish that have some tetrapod-like features, and then the first tetrapods that still have a lot of fish-like features and fins in certain places, and then finally we have full-blown tetrapods on land. The evolution of a horse. We have a pretty good fossil record of that. Let's see. From about 60 million years ago or a little older. And then through the Eocene, the Oligocene, the Miocene, the Pliocene. And of course, we finally have one species of horse nowadays. Oh, and we also, well, not really, we also have donkeys. Okay, so they're part of the same lineage. They've changed a lot. more primitive traits found earlier. The first horses were very small. Changes in tooth size, toe reduction. So a lot of these early horses had 5 digits. Eventually they were reduced to a single digit or hoof for each leg. Differences. Now, of course, we've also got some other, so there's some horses, there's also zebras, there's also asses or donkeys. I'm trying to think if there's anything else in that lineage. I don't think so. But we've got fossils from all these different sites. And then the only modern horse we actually have is the modern horse domesticated. It was thought that there's another horse called Brazilian's horse, which is actually a wild modern horse kind of thing, but recent evidence suggests that it's actually one of the first domestic horses that then went back and became wild again. So the key concepts that we've learned from horse evolution, fossil record demonstrates that evolutionary change has not been constant over time. There's been relatively long periods of relatively little change. For example, right here and right here, right here, things like that. And then sometimes it was a relatively rapid change, greater horse diversity in the past, much less later on. Some lineages evolve smaller body size, so it's always not always in the same direction. Multiple lineages exhibit similar evolutionary change. Okay, in terms of the fossil record, the first thing I want to point out, certain things fossilize well, like hard things, and other things don't fossilize well, like soft tissue, jelly, skin, that sort of thing. But we have fossils of both. Furthermore, most things don't fossilize. When an organism dies, it usually gets ground down to bits and there's nothing left of it. Only occasionally do you actually get a situation where a dead organism will actually form a fossil where minerals invade the tissue, replace the tissue, and you have mineralized form. Or you can have other kinds of fossils like impressions, things like that. So, then also fossils deteriorate over time, especially if they've been exposed. And just so you know, because of plate tectonics and the different plates that are moving around and subducting, there's actually very little rock left that has, like sedimentary rock, that was laid over by things falling on top of something and then turning into rock over long periods of time. There are very few rocks in that form that are older than 3 billion years. There are some, but not many, because there's this overturning of continental crust and these plates that they move along and then they subduct deep in the earth. They heat up and they're changed. It's called metamorphic rock. Or they get so hot that they eventually turn into molten magma. Okay? So anything that was in them is destroyed. And furthermore, erosion is a constant process. Whether it is in, let's say, rocks in a mountain, so there's weathering because of differences in temperature. There's weathering because of precipitation and water and snow. There's weathering from wind. And then, of course, there's gravity. So there are areas where billions of years of rock have disappeared. I'll give you a good example. In the Grand Canyon, at the rim, the topmost rock layer is, I think, I don't know, 100 million years old or something like that, 200 million. And then if you the further down you go, the older the rock gets, all the way to the Cambrian. It's called the tapetes, and 540 million years ago. And then there's also called the Great Discontinuity, where below that, at a certain level, there's 540 million years, and then rock that's 1.5 billion years. In other words, that intervening rock, it could have been mountains, it could have been all sorts of different things, has actually weathered away into fine grains and disappeared. Almost like it never was. And any fossils that were in there are gone as well. So forming fossils is very difficult. And just so you know, one of the complaints that a lot of people have about evolution is, oh, there's gaps in the fossil record. Well, of course there's going to be gaps in the fossil record, because the probability of actually forming a fossil, and then that fossil remaining intact, and then finding it, are so very small. But we still do have a fossil record. We even have fossil record of soft tissue. So this is actually a kind of a picture of Cambrian animals. These are metazoans on the seafloor and in the water column. All these things are, of course, extinct, but and a lot of these things have very soft bodies. But some beautiful examples of the Burgess Shale, this Cambrian deposit, have been found in British Columbia. And what happened was this whole seafloor and the animals that were living on it, underneath the sand, and then swimming around it, there was a giant slump or underwater landslide that covered everything up. And it stayed covered up for millions and millions and millions of years. It eventually turned into rock, and so it's actually been found, and most of these things are actually soft-bodied, so it can happen. Okay, so this is what I went over before about what kind of things fossilize. So I just want to recapitulate. We've gone through fossils. It turns out that evidence for evolution, even if there were no fossils, would still be equally strong. Fossils add a different dimension to it, obviously, but like I said, fossils are difficult to form. They degrade and weather over time and disappear. And so some things leave more fossils than others, especially like shelly organisms, with shells like clams and things like that, and bones and various things. Other things, even soft-bodied things, can form fossils, but they're uncommon. In the fossil record, we see things where we expect to see them, and we don't see things where we don't expect to see them. So in the last test, I mentioned that what evidence would suggest that descent from a common ancestor was not true. Well, if we found the remains of a modern human inside, above the pelvis of a Tyrannosaurus rex, like it had been eaten and had been fossilized together, that would kind of blow the roof off everything. So, of course, we don't see mammals until about 200 million years ago. If we found a mammal back in the Devonian 400 million years ago, we would have to rethink, perhaps start over in terms of theory of evolution. Okay. Now, comparative anatomy, we've talked about things like homology before, and we're going to continue to talk about those things right now by using comparative anatomy. So here we have all these different organisms with 4 limbs. We've got humans, we've got cats or felids, we've got bats. They look very different, but they still use the same limb bones. Here's a 5-digit limb hand with phalanges and such. And then you can even see some of that in whales where the fins have been, well, the things have been reduced, and now you have fins, but some of those same bones are still existent. So here we are with homology. Compare the same bones in different groups of animals. We have a humerus up here, and then here. We have a radius here, the ulna, 2 bones. Same thing in here. We have carpals, right? Actually, right here. Carpals. And then metacarpals. And then phalanges. Phalanges here, right? Metacarpals up here. A horse, we don't have phalanges that have all been fused into one bone. A porpoise with flippers, you can still see the 5 bones found in tetrapods of the hand and the foot. 1, 2, 3, 4, 5, right? But now it's been changed so that it's useful for swimming in water. So, different function and appearance, but derived from the same body part. This is homologous traits or homologous structures. So the traits might change in terms of whether it's a wing or an arm or something like that, but it's the same bones for slightly different functions depending on how this tetrapod actually makes its living. Now, what's the difference between homologous and convergent evolutionary traits? Remember, this is important, homologous traits are always traits inherited from a common ancestor. Whether it's the phalanges that we have on our hand or the phalanges that are seen in a bat's wing or a bird's wing. Okay? Now, there are convergent evolutionary traits. These are analogous traits. They look similar, but they are evolved independently and not due to common ancestry. Okay, so they're analogous traits. Example, the wings on birds, bats, and insects. In each case, wings evolved independently in each of these lineages. Insects much earlier. Birds and bats much later. I'm not sure how old. I think the earliest winged mammals were our bats, probably from the early Paleocene. Of course, the first birds are about 165 million years ago. Okay? So, convergent traits may look similar to homologous traits, but they evolve independently in unrelated taxa. So, if you're going to use wings as a trait to build a phylogeny, you very well might mislead yourself into thinking, well, perhaps wings only evolved once, and then inherited from, in all these different taxa, like insects, bats, and birds all had almost a common ancestor. But that would be misleading. They actually, winged flight evolved independently. First of all, mammals don't have wings or feathers. Birds do. Mammals have fur. Their wings are much different than the wings of birds. And then some insects have four wings and then lost a pair. And they're made from different structures entirely. So convergent traits, in other words, not descended from a common ancestor, can be phylogenetically. Okay. So here's more convergent evolution. Right here, we have a common ancestor, probably a shorebird with short legs. Okay? There's a split. One group goes to this side, and the other group goes to this side, right? I'll use this. Okay? And then, again, there's further bifurcation. You still have these two shorebirds. They're closely related to each other. They have short legs. You also have another short-legged individual. Let's see. Oops. Right here, right? But actually, this short-legged individual is more closely related to these long-legged individuals than these short-legged individuals. Okay? Because they actually have, this short-legged individual has derived traits that are shared by this big clade over here. And then finally there was an ancestor with long legs, initially, right there. And then these two then split into two different species. So long legs in this case is a homologous trait. It only arose independently once, and then subsequent descendants all inherited long legs. So the ancestral condition was short legs. Here we have analogous traits. Okay. Again, we have an ancestor for this whole thing. There are still two main clades. This clade and this clade. They share this common ancestor right down here. And then, finally, we have further splitting here to here to here. And to here. Okay. And then, finally, we have up here, this goes up to there, and we'll say this goes over to here. Okay? So, we have a long-tailed bird on the left. Now, if this was a homologous trait, like with a long-legged, then we would expect to see this bird and this bird to share a most recent common ancestor, but we don't. In fact, their most recent common ancestor is all the way down here, way in the evolutionary past. So it indicates that long tails evolved independently in these birds, and this is convergent evolution. It's misleading in terms of phylogeny. It's an analogous trait because of similar selective environments, but not descent from a common ancestor. So we ask, why is homology phylogenetically informative? Well, you have an ancestor, and descendants inherit the traits from that ancestor. If a new trait arises later on, then the descendants of the ancestor that first had those newly derived traits all have those traits, and the other ones might still have more primitive traits, like these shorebirds over here. Initially, they all had short legs, and then it was only later that a lineage, an ancestor had long legs, and then that further speciated. Long tails, on the other hand, evolved independently, perhaps from sexual selection, or perhaps ecology, similar environments, and not from a common ancestor. Okay, so some more examples real quick. These are plants that are adapted to arid environments where water is at a premium. Lack of water is what you have to get over. And so a lot of them can soak up water, like cactus in North and South America. There's a succulent in India that looks the same, but it's unrelated. But still the same sort of structure, and they can still suck up water when there is rain and store it. And then a euphorbia in Africa in a similar arid environment. Furthermore, this flower here, this is a carrion flower. It's red. It also gives off a stink like decaying flesh. Flies come in thinking it's carrion. They lay their eggs. They get pollen on them, and then they're attracted with that smell and the look of it to another carrion flower and pollinate that with the pollen from the other one. That you can find carrion flower plants in South America, in Africa, and in India as well. Okay? So those are also convergent, because in all cases you're actually deceiving insect pollinators like flies that thinking that it's actually carrion, lay their eggs, the larvae eat the decaying flesh, and then molt into an adult. Well, that's not the way it works. So convergent adaptations, again on feeding on nectar. We have 3 birds, all that feed on nectar. They have similar beaks that fit into the corolla of flowers, where the nectar is down at the bottom. Some of them hover. There's also an insect here. Doesn't have a beak. It has wings, but they're not. They're independently evolved wings, and it has a long proboscis that also fits into down the corolla of a flower. Now, a lot of birds are attracted to red flowers and things like that. A lot of moths come out at night for the most part, and a lot of those flowers are either light pink or white, but they all are very fragrant, whereas a lot of bird pollinated flowers are not fragrant, but they're brightly colored and with long corollas too. So again, distinguishing homology from convergence. Here we have a common dolphin. It's a mammal, right here. And then down here we have an ichthyosaur. It's actually a reptile, a marine reptile. It looks very similar. It's got fusiform body form, right? So this is convergence. They actually share a most recent common ancestor all the way back here. So we see this fusiform body form evolved independently, because this dolphin is also related to marsupials, monotremes, synapsids, primates, rodents, right? This whole lineage right here. And again, independently, these are reptiles, including lizards and snakes, ichthyosaurs, and then crocodiles, birds, and dinosaurs. They're not related to each other in terms of, but both these taxa and related taxa look that way because this is a very hydrodynamic anatomy that allows these animals to swim very efficiently through water. Homology. So, okay. Okay, so, hold it. Okay, so we have a common ancestor back here. First animals. Probably no Hox genes, perhaps a sponge or something. And then we finally get the beginnings of Hox genes. Humans and arthropods, like insects, they share a bunch of Hox genes that are all derived from a common ancestor. Okay? Back here. This one, this one, this one. And then genes in the Hox complex are fruit flies, right? So also here, and here, and here, and here. And then finally, this new Hox gene, but it turns out that there's only one of those for the fruit fly, but there's a bunch more for the primate and chordates and vertebrates. In other words, gene duplication. And Hox genes are kind of control genes that control expression, and other genes that control development from, let's say, an egg all the way up through an adult. Whereas the fusiform body form, mammals and ichthyosaurs actually were living at the same time, but obviously they're not related to each other, and they evolved that convergent form because of the environment they were in. The Hox genes are homologous. They're inherited from a common ancestor hundreds of millions of years ago. So, again, solving a similar problem in terms of flight, similar solution. We have everything from insects, birds, bats, and also pterosaurs. There were some vertebrate reptiles that also had wings. Convergent evolution, similar but not due to common ancestry. Again, so in this case we've got we have a niche, which could be burrowers, climbers, gliders. These are placental mammals like ourselves, right? And these are marsupials. A mole, it's a mammal, placental mammal. Can't see very well, but doesn't have to. We have a ring-tailed lemur that climbs very well. We have a spotted cuscus, among other things, that is a marsupial that climbs very well. Also like a koala, marsupial mole. And then a flying phalanger, also a marsupial. Okay? So, burrow, climb, glide. Same sorts of environments, same sorts of forms, because there's only so many solutions for each kind of different environment. So Australian marsupials evolved in isolation and not related to these placental mammals, but they evolved some of the same forms and functions that you found in placental mammals. Because Australia split off from Gondwanaland, about 250 million years ago. And at that time, there were marsupials, or the common ancestor of marsupials, but not placentals. They also have, in Australia, monotremes, or egg-laying mammals, the only place. So again, more anatomy sort of thing, but this is embryology or development. And all vertebrate embryos have gill pouch, right? Wherever you see the blue. They also all have tails, right? A chick. Eventually, in terms of this tortoise, they still have a tail, but the bird has lost its tail. Humans have lost their tails. Birds have lost their tails. A lot of reptiles still have tails. A lot of fish still have tails. Fish still have gills. Birds, reptiles, and mammals don't when they're adults, but when they're developing inside the womb, they are, in fact, they have gills. Vestigial traits are structures that with little or no apparent function, but resemble structures ancestors possessed. So, humans have muscles for wiggling their ears. We don't generally wiggle our ears much, but a lot of other animals do. Think of elephants. Deer. A bunch of different things can wiggle their ears. Boa constrictors have hip bones and tiny hind legs, even though they obviously don't need legs anymore. In fact, the legs get in the way of the way they move. Okay? They're fossorial, but they've been reduced, even though their ancestors had functional legs. These are no longer functional. Okay? Blind cavefish have small, non-functional eyes. So here we have an ancestor with an eye, and a cavefish where it's dark, and there's still the remnants of an eye, but it's not functional anymore. Why are humans prone to choking? Is it because of poor design? So there's this idea of an intelligent designer in a lot of Christian circles. In other words, a godlike individual that created things. And you would think that an intelligent designer would design things that would be as optimal as possible. But what we see here is that why are humans prone to choking? Okay? Well, if you had an optimal designer, you wouldn't design it this way, where you have one entrance for two different entryways, one for food and the other for air. It turns out that our ancestors were fish, of course. They had swim bladders. And in swim bladders, there really wasn't a danger of something going down the wrong side. Okay, they were used for buoyancy instead. But we still inherited all those same sorts of things, and so eventually, things that for air, let's say, actually we'll use this, went one way, and for food the other way, but they crossed. You would never do that. What you would normally do as an engineer is you dedicate one pipe to one thing and one pipe to the other. But evolution only works with what it has. It can't be optimal. It can only use what is already there and then selection will change things, but oftentimes you're still stuck with old things that worked in the past, like a swim bladder, but now you have a problem because the airway and the foodway cross, and sometimes food goes down the wrong way and you choke to death. Well, there's a lot of animals that are like this, right? You would think, why don't frogs choke? Well, part of it has to do with the fact that our larynx and pharynx actually descend when we get to be adults. Okay? And that's when we really have problems, where the airway and the trachea and the esophagus cross, and there's a valve there. It's actually a flap of skin that supposedly prevents things from going down into the airway, but it doesn't always work. The only thing that explains this is actual evolutionary history. Okay? Evolutionary history that we shared with fish with swim bladders, not lungs. So evolutionary history explains poor design. In other words, there wasn't a designer. Evolution by natural selection, but there were holdovers that are things that are now actually maladaptive, but we can't quite get rid of. And then the molecular record. Compare common genes and common proteins. Number of amino acids that are different from human hemoglobin. So, macaque, there's 8 amino acid changes between humans and macaques, and we're much more closely related to each other than we are to, let's say, birds or lampreys. Human hemoglobin compared to dogs, so we shared a more recent common acid with macaques up here, right? This is all, again, time. Younger, older, older, younger, right? And we go further back in time. Here is where we share our most recent common ancestor between dogs, right there, and humans. And that's 32 different amino acid changes in the hemoglobin. If we go back further in time to birds, there's now 45 different changes in amino acids for our hemoglobin. Frogs, 67, either further back in time, right? And then finally, lampreys, really far back in time. There's 125 different amino acid differences in human hemoglobin compared to lampreys. This is what you would expect. And underlying these amino acid changes are, of course, changes in the DNA that code for amino acids, proteins. And then finally, we have instances where we can actually see evolution in action. In fact, one of the things that Darwin noticed, of course, he was part of the landed gentry, and a gentleman didn't have to work. So one of his hobbies was breeding. He bred pigeons, and he also bred some plants. So he used artificial selection, and he was very familiar growing up with selective breeding. The English were very much into it, especially with dogs and agricultural animals and plants and all that kind of stuff. And they would breed for certain traits. They would select for certain traits. In other words, you have animals that, let's say you want a cow that produces lots of milk. So you would breed, you have individuals that are born. Some produce more milk than others. You take the ones that produce lots of milk and breed them with other individuals that also produce lots of milk. And over time, you're going to get a cow that produces an extra large amount of milk. Similarly, so this is corn. The wild plant is actually teosinte. The kernels are very small. They were harvested wild, and then eventually Mesoamericans in the central Mexican highlands, they started selectively breeding them. Instead of just harvesting them in the wild, they started, perhaps some dropped near their villages, they started growing, then they started actually growing them on purpose, and then when they always took the plants with the largest kernels, planted those kernels, and selectively bred from individuals that produced larger and larger and larger kernels until we now have modern corn. Okay? And you can find all sorts of intermediates along the way. If you go into the southwest United States or into Mexico, you see all kinds of different corn, oftentimes with different colored kernels, things like that. We even do genetically modified. So, artificial selection, again, evolution in action. So, here we have a wolf. Wolves are wild animals. The closest relatives to wolves are actually domesticated dogs. The thing is, look at all these different kinds of dogs. We have some dogs that are huge, like the Saint Bernard. We have sausage dogs, like this Dachshund. We have more wolf-like dogs, like here. We have dogs of different sizes, smaller dogs, right? In fact, we actually have some little yappers down here, like Chihuahuas, that are basically genetic monstrosities. They were all bred for certain traits and certain lifestyles. Okay, so Saint Bernards were bred for living in the Swiss Alps, where the weather was very cold, and they often were used for, with a shepherd. Long, slender dogs were often used for going into holes and caves and things like that. This black lab was bred for retrieving, usually game that has been shot or to capture it. Okay. So over time we created all these different breeds. Some of the breeds we just created just because we wanted certain traits that not necessarily were any good or not. They just looked kind of cool. Okay. Well, it turns out in the former Soviet Union, there was this Russian geneticist. A lot of people in Siberia were actually raising silver foxes for fur. And of course, silver foxes, they're wild. They don't much like humans. They're pretty aggressive and nasty towards humans when they're captured and all that. And they wouldn't make necessarily a very good pet. Okay? But this guy, what's his name? I forget. He's dead now. And his former assistant, she's still alive. They started breeding silver foxes to see what would happen if they just selected for one characteristic, and that's called tame, being like domesticated, having a nice personality, actually like being around humans. And within about 50 generations, and they could produce quite a few generations per year, so over about a 25-year period, they selected for these silver foxes that became more and more and more tame and actually seemed to enjoy being around human beings and liking attention from human beings. What was also interesting is that as these foxes evolved for tameness, their ears started to droop, their tails started to curl, and they started getting spots on them like white blotches and all that you see in domestic dogs. And it seems like when you select for tameness, there's a whole bunch of other traits that are pulled along. It's called correlated evolution. And so nowadays, this program is still going on. It's actually being funded more by Americans, but it's amazing that over such a short period of time of artificial selection, you basically have a domesticated fox. So, in terms of artificial selection, the evidence of evolution, the same processes that act within populations, can act over time to lead to new species, new biological diversity, new branches on the tree of life. Okay? So, instead of us selecting, it's actually the environment that selects, but the environment is not an entity. But still, certain forms and functions operate or are more efficient, more beneficial in some environments than in others. And so you get the same sort of evolutionary change and adaptation. And adaptation only comes from evolution by natural selection, just like artificial selection. So we have bred for modern corn with big, juicy kernels. Okay? In the same way that an environment might breed for, let's say, a long curved beak for drinking nectar. So, we have a question. Can artificial selection lead to substantial evolutionary change? Strong directional selection will quickly lead to a large shift in the mean value of a population. In one population, every generation, pick out the 20% of the population with the most bristles and allow them to reproduce for the next generation. And for the other population, do the same with the fewest. And what will we eventually see over time? We will actually see populations with many bristles, even though beyond what the initial range was. That's the initial range between 30 and 50. And then also with fewer bristles. Right? And so now we've got a range of almost no bristles all the way up to over 110. Even though originally there was not enough variation that you could in any way actually get more than what the original range was. But artificial selection, there's always mutations, and eventually there'll be mutations that give you some more bristles. And so you just, over time, breed flies with many more bristles, and eventually you'll get many, many more bristles on flies or many fewer. So, evolution in action. This is actually fairly rapid evolutionary change due to natural selection. One of the best, well, it's kind of classic study. During the Industrial Revolution in Europe and England, they burned lots of coal for manufacturing, and then of course people burn coal for heating and things like that. And over time, a lot of coal soot was released, into the atmosphere and coated everything, including the trees. And so previously, we had trees with white bark that, like on the left, and now we had trees that were really dark on the right. But as you can see, there were two forms of moths, of peppered moths. One form was most common before the use of coal, because the bark was kind of mostly white and some darkness. And so, as you can see, it was camouflaged perfectly, completely cryptic. Now, if we took that one and put it over here, it would obviously be visible. And so that frequency, as more and more trees got covered, the frequency of the light moth decreased, and the frequency of the dark moth increased, corresponding to selection on survival by birds. So birds couldn't see the dark morph. Now, prior to the industrialization, there were dark morphs, but they were rare for the most part. And then here, you can see the same thing with some lizards. Okay. Now, so here we have, I want to point out, prevailing wind. Okay. We have light, and then we have dark, right? And in places where there was little industrialization, we see the white morph, like in Ireland, and then up in Scotland as well. But most of the industrialization, by the way, was down in southern England. And since the wind blew that way, the soot was carried north. And as you can see, there are no pollution controls or anything like that. So this was the state of affairs for, beginning in the late 18th century, early 19th century. And it got worse and worse and worse. However, and this study was done by this guy, Kettlewell, and what he did was he took all these moths, light ones and dark ones, and released them, and then tried to recapture them when the bark was dark. And of course, the dark morphs, there were a lot more of those, and very few light morphs. But then things started to change with pollution controls in the 70s, and things got cleaner and cleaner and cleaner, and now the light morph is becoming more common, in other words, higher in frequency, because there's no longer tree bark that's being covered by coal dust. So, pesticide resistance, very much similar to antibiotic resistance. In this case, we have wild type. here, 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 here. But there's a mutant then right here. So the wild types, all of them go down. But the mutant survives and produces offspring. And they produce offspring. And eventually, we have lots of offspring. And pesticide was DDT mainly back then. It had a terrible effect on the environment, killed lots of birds, and it also made their eggshells very thin, which led to reproductive failure. Here, antibiotic resistance in Staph aureus, getting very common in hospitals. I'm not sure yet, but I'll probably have you guys upload this. So you'll have to fill in each of these in terms of with those A through H. So in terms of some misconceptions, remember selection is not always for the biggest and strongest. If you remember jack salmon, the small salmon that only stay a year in the ocean compared to the normal adult salmon that go out for 3 years and come back in their 4th year to spawn. When jacks are rare, they actually have higher fitness than large males when they're common. Okay, so just remember it has nothing to do with size. Or sometimes it does have to do with size, but for smaller size. Organisms are only adapted to the current environment and current utility and not to any future. Even though, it'd be nice if, we could eventually have humans who could fly, but it doesn't look like it's going to happen. Organisms are not optimally designed or perfect. They're actually good enough. If those traits that are good enough lead to increased survival and reproduction, then they don't have to be perfect. They just have to be functional. A good example, also, evolution only works with what is available. In other words, standing genetic variation in the population. And so it doesn't always lead to the best outcome. A panda has 5 fingers, but like most bears, are 5 in their paws, 5 digits. But they're really they're designed for running, or they function for helping them run. And that's from their evolutionary past, but they also have a sesamoid bone right in their wrist that has, over hundreds of thousands of years, evolved into like a 6th digit, like a thumb, that they use to grab bamboo. It's not nearly as functional a thumb as ours, but it works well enough. Evolution also, by natural selection, is not random. Okay, so evolution, critics and the critics of evolution. Evolution is not demonstrated. Well, scientists use the word theory differently than popular culture, just like the theory of gravity is based on many years of experiments and and our works in progress. As with all disciplines, we are learning, but we can apply the theory of evolution to understand the history of life, the patterns of lineage splitting, and the mechanisms of change. This is how we understand antibiotic resistant works. Why are there physical matches between pollinators and flowers? Evolution is the only well-supported explanation for the diversity of life on Earth. I gave a long spiel about the fossil record and how difficult it is to make fossils, and even when you make fossils, the fact that they erode with everything else, and so most fossils are eventually lost. Also, subduction zones and all that. So of course there will be gaps. The fact that we do have pretty decent fossil record for some lineages over, evolutionary time, millions of years, certainly shows support for that. But that doesn't constitute evidence against evolution. In fact, that's exactly what you'd expect. We would expect to see transitional forms. We'd see homology. Not all organisms fossilized, so it's apt to be some gaps in the record. If you want to go into this deeper, go to Understanding Evolution at the Berkeley website below.